Welcome to Jinnious Lab. Today we will cover IB Biology B1.1.1-7 Lipids.
B1.1.8 to 11 is our chapter is about lipids. So first, let's learn about the types of lipids and their properties. This is a diagram of lipid categorization into fatty acids, steroids, the uh, and the triglycerides, and lots of other categorizations under the lipids category. Lipids are nonpolar and hydrophobic hydrocarbon chains, such as fatty acids, or rings, such as steroids. And with the conception of like dissolves like, long hydrocarbon chains that are nonpolar have low interaction with polar water, so it has a really low solubility in water because lipids are hydrophobic. However, it has high solubility in nonpolar solvents because both of them are nonpolar. However, there are amphipathic lipids that include phospholipid, triglyceride, and cholesterol. These three are amphipathic lipids, and what amphipathic means is that it contains both a hydrophobic and hydrophilic regions. In the case for phospholipid, the phospho, which is the phosphate head, is the one that is hydrophilic, and the lipid part is the hydrophobic part. For cholesterol, there is a polar hydroxyl group with the lipid, which is the hydrophobic one. And for the glycolipids, there's a polar carbohydrate chains and a non-polar lipid. Now, for the transportation of hydrophobic lipids, it's important to notice that in the case of polar materials, they can be dissolved in the blood plasma and be transported throughout our body easily because they can be dissolved. However, in the case for lipids, since they're non-polar and since they cannot be dissolved in our body and our blood plasma, it needs to be transported via alternative method. And this is done through the lipoprotein, the packaging of certain lipid or this hydrophobic lipid with a lipoprotein that allows the packaging of with a protein coat that is hydrophilic. Now, the lipoprotein contains a lipid and a protein vesicle that carries fats and cholesterols to regulate the blood cholesterol levels by moving and transporting the fats and cholesterols in our blood. Now, the surface monolayer of the phospholipids and the free cholesterol um, is the hydrophilic region, which allows the transportation around our body. While the outside layer is hydrophilic, the inner layer is hydrophobic core, and this allows the packaging and the interaction with hydrophobic lipids. It is similar to the structure of egg, where the A, which is the egg white A, is the phospholipid monolayer with the hydrophilic phosphate head facing towards the outside for the ease of transport, and the egg yolk B, where the hydrophobic lipid is wrapped with the egg white, which is the phospholipid monolayer. Now, the lipoproteins have more to learn about. There are two types of lipoproteins. There's low density lipoprotein and high density lipoprotein. In the case of low density lipoprotein, they are typically called as the bad cholesterol because they carry the cholesterol from the liver to the body to disperse the cholesterol. And as the cholesterol is delivered from the liver to our body, which can be anywhere, any areas of our body, the cholesterol level increases in our blood, and uh, this can cause adverse health effects in our body, uh, such as shocks or blood clotting. And this LDL can be derived from eating trans fats and saturated fat that uh, that increases the LDL level. And these trans fats and saturated fats are typically found in fried foods, butter, full fat dairy, red meats, such as beef. And for high density lipoproteins, it's typically called the good cholesterol, which scavenges the excess cholesterol from the body to the liver for disposal. This decreases the blood cholesterol level, which balances out the increasing blood cholesterol level due to the LDL. That's why it is important for us to always eat the cis fat, which increases the HDL level. And the cis fat is abundant in avocado, olive oil, and oatmeal. A second type of transportation of hydrophobic lipids is thyroid hormones, where it's bound to carrier proteins to facilitate movement. Now let's go through the lipid functions. For the lipid functions, there are mainly three functions, which is first, energy storage, where the plants store the fats or oils in their seeds primarily as unsaturated fats in order to use the energy in germination and seedling to grow and establish itself until it is capable of photosynthesis. The second function of lipid is thermal insulation, where the endotherms, which is simply animals that generate heat to maintain constant internal body temperature, to store triglycerides in the adipocytes for supply of the energy. For example, the whale bubbler, whale has they have a bubbler which is composed of adipose tissues that contain large amounts of triglycerides. And whenever they need to increase their temperature, they can release the energy from the triglycerides that they are stored and increase their body temperature. And this is their way of thermal insulating them, even in cold temperatures of the ocean. The function of lipid is related to its hydrophobic nature. For instance, in plants, the waxes prevent the water loss from the leaves, and for animals, it coats the bird's feathers with oil to make them waterproof. So its hydrophobic nature allows plants to prevent water loss and animals to have waterproof wings. In the case of phospholipids, it forms spontaneous membranes in aqueous solutions. In the case of lipids in foods, it supports absorption of fat-soluble micronutrients such as vitamin A, D, E, and K. Now, for the second type of lipid, let's look at the triglycerides and phospholipids. Triglycerides and phospholipids are formed by condensation reaction and are linked by ester bond. Since we've went through condensation multiple times, let's just quickly view how the condensation reaction forms the triglyceride. So triglyceride is composed of one glycerol head and three fatty acids. As you can see, this is one glycerol head, and this is an example fatty acid. There must be one, two more. So in total, there must be three fatty acids. But as you can see, H2O is released as a byproduct, and the COC and the double bond O up here forms the ester bond. This ester bond is formed with three fatty acid tails with the glycerol head and forms the triglyceride lipid. This is for long-term energy storage. In the case of animals, it's stored as fats. For plants, it's stored as oil. The phospholipid is composed of one glycerol and one phosphate group with two fatty acids. This is basically the same thing that you've drawn in phospholipid bilayer, where you can see that the phosphate head is here and the fatty acid the phosphate head is here and the fatty acid tails are here. So it is the triglycerides are modified, and the third fatty acid can be replaced by the phosphate group. So basically from this, it is modified to this, as one of the fatty acid tail is replaced with the phosphate group. It is a major component of the cell membranes. Now, let's move on to the fatty acid chapter, where we would like to learn about the three major types of fatty acids. There are saturated, monounsaturated, and polyunsaturated. And this categorization depends on the number of double bonds between the carbon-carbon. As you can see, saturated fatty acids have zero double bond, which allows them to have a linear structure. In the case of monounsaturated fatty acid, it contains one carbon-carbon double bond. You can memorize this by the prefix mono, meaning one. And in the case of polyunsaturated fatty acid, it has multiple double bonds between the carbons. And you can memorize this by how poly prefix means some. Because there are no double bonds in saturated fatty acid, the structure is linear. However, for the mono and polyunsaturated fatty acids, because of the double bond, there are kinks. However, for the monounsaturated one, there's only one double bond, therefore there's only a slight kink. And on the other hand, for polyunsaturated, there are greater degree of kink that's present. This is visible in the diagram here, where the saturated fatty acid has a linear form, while the monounsaturated fatty acid has a slightly kinked structure, and how the polyunsaturated fatty acid contains a greater degree of kink in a structure. Now, there are isomers of unsaturated fatty acids, which are cis and trans. Cis fatty acids are the ones that have this when the hydrogen atoms are on the same side of the carbon-carbon double bond. And trans is when the hydrogen atoms are on the opposite sides of the double bond. And to help you memorize which one is cis and which one is trans, you can see how when the hydrogen atoms are on the same side, it resembles a C that is 90 degrees flipped. In the case of trans, you can see the T that is present when the hydrogen atoms are on the opposite sides of the double bond. Now we would like to compare between the trans and saturated fatty acids. So as I've mentioned before, low density lipoprotein and high density lipoproteins level and abundance alters depending on the consumption of trans, saturated, or cis fat. For trans fat, it increases LDL and decreases HDL. And you should remember that LDL brings adverse health effects, which is also the reason why it's typically called the bad cholesterol. And HDL contains good cholesterol. So the increase of bad cholesterol and decrease of good cholesterol results in the worst form of fat. It is found in cookies, donuts, cakes, and fries, and it's not common in nature. It's typically produced by industrial processes, which is hydrogenation, and it's solid in room temperature. The bad type of fat is the saturated fat, which increases the LDL but doesn't decrease the HDL. Food examples include beef, butter, pizza, and ice cream. For good fat, it's the cis unsaturated fat, which increases the HDL. It involves almonds, vegetables, fish, and olive oil. It is common in nature and it exists as liquid in room temperature. For LDL and HDL and their health risks, we would like to learn a bit more here. So fats are insoluble in cholesterol, and hepatic is only slightly soluble. They are carried in blood as tiny lipoprotein particles and high-density lipoprotein and low-density lipoprotein. For LDL, it stores cholesterol in the bloodstream, while the HDL removes the cholesterol from the bloodstream. As high level of LDL in the bloodstream increases the risk of coronary heart disease due to atherosclerosis, to become healthy, you're supposed to take cis fat and do not take trans fat and saturated fats. Now, moving on, let's learn about the phospholipid bilayer. And phospholipids form the cell membrane that encloses and protects the cell. So when you draw the cell structure, you would always draw the phospholipid bilayer that encloses all the organelles and the cytoplasm. It is an amphipathic layer that consists of hydrophilic with negatively charged phosphate head and hydrophobic nonpolar fatty acid chains. Due to its amphipathic nature, the phospholipid molecules form a special structure called the phospholipid bilayer. Here you can see that the phosphate and glycerol head is hydrophilic, and how the two fatty acids that are linked with the phosphate head are hydrophobic. And this is the reason why the bilayer exists in this form, where the hydrophilic heads are facing either the intracellular region or the extracellular region, while the fatty acid tails, which are hydrophobic, are attracted to each other. Now we would like to learn about steroids. Steroids are naturally occurring hormones that are composed of four carbon rings. As they are hydrophobic, they pass through the phospholipid bilayer faster in response for the signal transduction. For example, it includes cholesterol, testosterone, which is the male reproductive development hormone, and the estradiol, which is the female reproductive development hormone. The steroid skeleton is like the Harry Potter's mark in his forehead, where the three hexagons are attached to each other, and a pentagon is attached on the top right. And you can see the thunder type of shape in the steroid skeleton. So examples as mentioned here include cholesterol, testosterone, and estradiol. And I'm sure that you wouldn't have to memorize the whole structures, but you should know how to identify which one resembles which cholesterol type. Now let's compare the carbohydrates and lipids as energy storage. For carbohydrates, it contains 4 calories per gram, while lipids contain 9 calories per gram, which is more than twice of the carbohydrates' energy storage. Therefore, lipids are more efficient energy storage, and the storage efficiency increases because lipids are hydrophobic and less mass is taken up storing water. Carbohydrates are easily synthesized for storage, and they are easily broken down to release energy quickly, such as glycogen in animals, starch in plants. However, lipids are complex in synthesis and breaking down is very slow. So, as a way for you to memorize, carbohydrates are like wallets. You can easily take out your money. However, you can only keep limited amount of money in your wallet. For lipids, it's like your bank account where you would have to visit the bank to take out all your money. So you would have to go through a more complex breaking down process. However, you can store more money. You can store more energy in the storage method. In the comparison of fats and oils, you can compare how the fats are a type of storage for endotherms, which is basically animals, and how the fatty acids that are stored are saturated, which has the linear straight chain structure, and that in room temperature, it stayed as solid. The melting point is comparatively high, and in the case for oils, it's found in plants and it's stored in unsaturated cis structure with kinked chains. And for its state in room temperature, it's liquid and the melting point is comparatively low. In the case for body mass index, this is a way for you to calculate whether your body mass index is underweight, normal, overweight, or obese. Here is the chart to calculate the body mass index. And here's a toggle for you to practice example past paper question after referring to how to calculate the BMI with this calculation bar. And there is a mark scheme attached for you to check if your answer is right or not. 한 거예요.
잘했어, 잘했어. 매우 잘했습니다. 아주 잘했어요. 그러면 언니도 한번 해볼게요.
네.
잠깐만. 음,
For IB45 Genius, Genius Lab, don't forget to subscribe and like.